All right, folks, let's talk about what you need to study for your unit assessment on DNA. We have two foldables for this unit, one that is about the history and structure of DNA and the other that is about mutations. In your labs and projects section, you have two different lab sheets. We have the Intro to DNA Web Quest and the summative, the DNA molecule. I gave that back to you if you turned it in on time. We will be putting that in our portfolios, but hopefully you still have that. The back is a great study guide. You also have your unit portfolio for our DNA unit, as well as your DNA glossary. And as always, if you go onto Student View, Parent View, Phoenix, you can find full copies of notes and class presentations, review games, as well as links to this and other helpful review videos. All right, so what exactly is DNA? DNA, or deoxyribonucleic acid, is a molecule that's inside all living things that acts as a blueprint for life. It tells cells how to grow, how to develop, how to reproduce, how to make proteins. It tells them everything they need to know in their lives. It's a double helix or two-stranded twist shape, and it's the key to all successful cell life. Different kinds of cells will have their DNA in different parts of cells. Prokaryotes, like archaea and bacteria, don't have a nucleus, so their DNA is a big tangle found in the cytoplasm. Eukaryotes, or cells with nuclei, which include fungi, protists, plants, and animals, they have their DNA inside the nucleus. Last unit we learned about how cells copy their nuclei in a process called mitosis. Most of the time though, we don't see the DNA in those X-shaped chromosomes. Most of the time it's just hanging out in a decondensed, untangled state. DNA is the molecule that makes us all unique. No one else in the entire world who has ever lived before or will ever live or is living now has your exact same DNA. That is, of course, unless you have an identical twin. Or maybe
an evil twin.
Let's talk a little bit about the history of DNA. Even though DNA science is a relatively recent field of study, as in the structure of DNA has been deciphered only within the lifetimes of our grandparents and great-grandparents, people have wondered about heredity and how traits were passed on through generations throughout all of history. Even without microscopes and the internet and Google and all sorts of advanced imaging sciences, people way back in the day knew that they could breed certain animals, such as wolves, for certain desirable traits, such as loyalty or friendliness, and eventually create new species, such as our favorite, the domesticated dog. People also noticed patterns of inheritance in the offspring and parents of animals and people around them. Some, like Gregor Mendel, we'll learn more about him next unit, researched plants and observed this inheritance of traits from generation to generation. In this unit, we learned about several influential scientists whose work helped us learn the structure and function of DNA. Erwin Chargaff, who suggested through his research that there was a relationship between the amount of adenine and thymine and guanine and cytosine in any sample of DNA. Rosalind Franklin, whose famous image of a crystal of DNA called Photo 51 revealed the double helix structure of this fantastic molecule. And Maurice Wilkins, James Watson, and Francis Crick, who would go on to build the first model of DNA using the research of Chargaff and Franklin without necessarily giving them the credit that was due to them. Once upon a time, there were some scientists: Maurice Wilkins, James Watson, and Francis Crick, who are all interested in cracking the code of DNA and figuring out how to best model this mysterious, important molecule. Although these four scientists did not get along personally, they cooperated professionally and collaborated by sharing data. Thanks to Chargaff, these three scientists were able to build their model, for they suggested this pairing mechanism between the four bases. Now, these scientists knew that there was some kind of connection between the amount of guanine and cytosine and adenine and thymine in any sample of DNA, but they weren't quite sure how they paired up together. Their initial model was actually kind of inside out. We know today that there are phosphates and sugars on the outside and that these base pairs match up on the inside. but these scientists did not know that at the time. It was not until they had the help of Rosalind Franklin, specifically photo 51, that they knew that they were dealing with a double helix structure. This photo was borrowed without permission by Maurice Wilkins and used by this team of scientists to build their model. Franklin would go to her grave without realizing the impact that this photo had on creating the first model of DNA. Watson, Crick, and Wilkins would go on to win the Nobel Prize in Physiology and Medicine for their model of DNA. They did not give fair credit to Franklin and Chargaff, which is really unfair. So now let's talk about this structure that they uncovered. DNA, as we said before, is a double helix, or a two-stranded twist. And if we were to think of this structure, this double helix, like a twisted ladder, the sides of the ladder would be the phosphate sugar backbone. And the rungs of the ladder, what you might climb up if you were using this to get from A to B, are the base pairs. The base pairs provide information and make every organism unique. If you want to build a molecule of DNA, you're going to need a lot of nucleotides. Nucleotides are like the bricks in the building that is DNA. Each nucleotide consists of a phosphate group, we used black beads in our model for that, a sugar called deoxyribose sugar, and a nitrogenous base of your choice. And a nitrogenous base, we used the colorful beads to represent the nitrogenous bases, and in case you forgot, they pair up like that. Because of these, any nitrogenous base you choose can go in there with the deoxyribose sugar and the phosphate group to create what's known as a nucleotide. Nucleotides are like the bricks in the building that is DNA. These nucleotides line up together to make the amazing molecule known as DNA. These four letters, A, T, G, and C, make up all life on Earth. Through infinite combinations, you can create all kinds of organisms, proteins, and molecules. Here we have part of a molecule of DNA. You'll notice the base pairs are in the middle. The deoxyribose sugar and the phosphate groups are on the side, forming that phosphate sugar backbone. You might have also noticed that the phosphate groups, represented here by black dots, are pointing in opposite directions. This is because the strands are said to be in anti-parallel, meaning they're not running in exactly the same direction. I use the example in class if it's kind of like a disco person dancing, pointing their fingers in opposite directions. If the cell gets the signal that it's time to copy its DNA, this strand of DNA will split apart. Each old strand will serve as a template to form a new strand. The result will be two new strands, each containing 1/2 old and 1/2 new material. The new material will match up perfectly following Chargaff's rules, and the new base pairs will be created using substances synthesized by the cell or perhaps ingested as food. So you've got your corresponding base pairs. The resulting strand is exactly like the old one in anti-parallel. Now we'll do the other side. As you can see, now we have two identical copies of the original strand of DNA. When all of this is happening, sometimes stuff goes wrong. Just like when you're texting a friend or typing up a paper, sometimes you make a typo. When we have these typos happen, this is called mutation. Now, mutation and mutations get a really, really bad reputation, which is kind of unfair. Sometimes we think of them causing superheroes. Other times we think of mutations causing some kind of terrible, ugly disaster. But in reality, a mutation is just a change in DNA. The word mutation comes from the Latin word that means I change. So mutation is just a change. We learned about 4 different kinds of mutations in this unit. We'll go over each one using an example starting original strand. So if my original strand of DNA looks like this, and it gets copied with a mutation, the message or the code in the DNA is going to change. So let's start with an insertion mutation. If an insertion mutation happens, that means an extra nucleotide is added where it wasn't before. So my original strand says this, but my copied strand might say, notice how this extra adenine was added or inserted. A deletion mutation is a lot like what it sounds like. It means that one of the original nucleotides gets deleted or removed. Perhaps the new strand might look like this. Notice that I'm missing that thymine that was in the original strand. It got deleted in my new strand. Other times we might see a substitution mutation. Just like if I'm out sick, I have a substitute coming in to replace me to teach class. A substitution mutation is when one original nucleotide just gets swapped out for another. Notice how instead of a thymine, I now have a guanine in this copied strand. That's going to definitely change the message. And the last kind of mutation that we learned about this unit is an inversion mutation. This is when two original nucleotides in a sequence just trade places. Notice how my adenine and thymine switched places in the copied strand. Because all of these mutations change the original message, the new DNA might say something different than originally intended. Sometimes this can be good, sometimes this can be bad, and sometimes this might have no effect at all.
A always pairs with T, G always pairs with C. These are our base pairs. They're on the inside of this molecule of DNA, and they're the code that is the blueprint of life. The infinite ways that you could combine these 4 base pairs gives you all the variety of life on Earth. To build this molecule called DNA, you need to have a nucleotide. A nucleotide is made up of a phosphate group, deoxyribose sugar, and a nitrogenous base of your choice. These together give you a nucleotide. For our scientists, we need to know Rosalind Franklin is famous for making photo 51. Watson, Crick, and Wilkins, Nobel Prize for their model of DNA. Erwin Chargaff, who suggested through his research that there was a relationship between the amount of adenine and thymine and guanine and cytosine in any sample of DNA. So here's a strand of DNA. We have one side and the other. They are complementary strands to each other. They complete each other. If the cell gets the signal that it's time to copy its DNA, this strand of DNA will split apart. Each old strand will serve as a template to form a new strand. The result will be 2 new strands, each containing 1/2 old and 1/2 new material. The new material will match up perfectly following Chargaff's rules, and the new base pairs will be created using substances synthesized by the cell or perhaps ingested as food. As you can see, now we have 2 identical copies. When all of this is happening, sometimes stuff goes wrong. Sometimes you make a typo. When we have these typos happen, this is called mutation. A mutation is just a change in DNA. We learned about 4 different kinds of mutations in this unit. So if my original strand of DNA looks like this, and it gets copied with a mutation, the message or the code in the DNA is going to change. If an insertion mutation happens, that means an extra nucleotide is added where it wasn't before. A deletion mutation is a lot like what it sounds like. It means that one of the original nucleotides gets deleted or removed. Other times we might see a substitution mutation. A substitution mutation is when one original nucleotide just gets swapped out for another. And the last kind of mutation that we learned about this unit is an inversion mutation. This is when 2 original nucleotides in a sequence just trade places.